Okay, so alpha oxidation is happening on those fatty acids which are having a side chain on the beta position. So since there is a presence of side chain on the beta carbon atom, the beta oxidative enzymes cannot come and act in between the alpha and the beta. So what happens? There is some modification happening on the pathway and removes the alpha carbon atom. So the alpha it's not the alpha carbon, it's acting on the alpha bond that is in between the first and the second carbon atom. So such an oxidative pathway is known as the alpha oxidation and something different from that of the beta oxidation. The initial steps are something different, right? That is the pathway, so that the pathway is modified in such a way that this carbon dioxide will be removed so that the side chain now shifts to this carbon dioxide will be removed and this carbon is oxidated to COO minus, right? So this COO minus, so this forms the first carbon. When it becomes COO minus, this becomes the first, this becomes the second, and this whatever CH2 tricycle or CH2 becomes the third carbon atom. So now this bond is free. It is away, the beta oxidative enzymes can act, right? So the thing happening is the removal of carbon dioxide or COO minus from the terminal position, right? So that the number is reduced by 1. So if phytanic acid is a C20 fatty acid, so when the alpha oxidation happens, what happens? This carbon dioxide is removed in the form of carbon dioxide or formic acid, right? So that it is now converted into an odd numbered fatty acid, right? Once this odd numbered fatty acid is formed, it can then follow the beta oxidation. Odd numbered fatty acid, and again it is a very long chain fatty acid, so it's the beta oxidation is not happening. Once the alpha oxidation is over, the beta oxidation is not happening on the mitochondrial matrix, but instead it will be happening in the peroxisomes because it is VLCFA, very long chain fatty acid. This very long chain fatty acid has to be first converted into a long chain fatty acid, then only the C8 octanoyl CoA indication it will be entering into the mitochondrial matrix and the beta oxidation will be continued, right? So there are some modifications happening. Beta oxidation and the simple enzymes one fatty acid oxidation will be completed. Then we discussed about what happens in the odd chain fatty acids. Odd chain fatty acids the reaction happening is the same, but the end product of the last cycle will be propionyl CoA rather than acetyl CoA, right? So that was the beta oxidation of odd chain fatty acid. Then we studied a slight modification of the beta oxidation, which is known as the peroxisomal oxidation, which is happening in the peroxisomes rather than the mitochondrial matrix, where we are dealing with the oxidation of very long chain fatty acids. So the very long chain fatty acid is first reduced to octanoyl CoA, then the usual mitochondrial beta oxidation will be taking place, right? Now we are studying another modification of beta oxidation or another a variant of beta oxidation, which is known as alpha oxidation, which is happening only on that fatty acids which are having side chains on the beta carbon atom. So if you are having if you are consuming more and more cow's milk, or you are strict vegetarian, you'll be having phytols in your diet, right? Because chlorophyll is rich in phytols. So this phytol it will be converted into phytanic acid, so which now behaves as a fatty acid. So this phytanic acid cannot be processed by the normal beta oxidation because the beta oxidative enzymes cannot, or they are prevented from the action with the side chain present on the beta carbon. So instead of going into the beta oxidation first, it first carry out the alpha oxidation, then it follows the beta oxidation. Clear? എന്തായാലും beta oxidation finally എത്തും, പക്ഷേ the fatty acid has to be modified first, right? So how is it modified? The phytanic acid comes, right? It is first undergoing hydroxylation reaction, right? Hydroxylation reaction. What do you mean by hydroxylation? That is hydroxyl group. OH group is added where this CH2 is converted into CHOH. So the OH is attached to here, right? So what happens to that H? H can be attached to this O minus. Clear? So CHOH, or simply we write it as CO minus because it is not the hydration happening. HOH, water molecule. Water molecule as such is not coming, but water molecule is split in the form of OH, and this OH is only attached to the phytanic acid, so that it is now converted into 2-hydroxy. This is known as 2-hydroxy or alpha hydroxy phytanic acid, right? So hydroxylation happens first, introducing an OH on the alpha position or the second position, so that it is known as alpha hydroxyphytanic acid or 2-hydroxyphytanic acid. Now this 2-hydroxyphytanic acid undergoes decarboxylation. Clear? So what happens during decarboxylation? This carbon dioxide will be removed by decarboxylase enzyme, or it is also known as phytanic acid alpha oxidase. എന്തുകൊണ്ടാണ് അതിനെ ആൽഫ ഓക്സിഡേസ് എന്ന് പറയുന്നത്? കാർബൺ ഡൈ ഓക്സൈഡും പോകുന്നുണ്ട്, ഒപ്പം തന്നെ ഈ രണ്ട് ഹൈഡ്രജൻ ആറ്റവും പോകും. Right? So it is known as COO minus. Right? Oxidative decarboxylation is happening. So it is the carbon atom, the number of the carbon atoms is reduced by 1. So C20, that now we are having C19 with the removal of the carbon dioxide and the alpha carbon atom, or now this carbon atom is converted into COO minus. Clear? This product is known as pristanic acid. This is known as pristanic acid. It's an odd chain fatty acid. Clear? Now it is converted, pristanic acid is converted into pristanoyl CoA. Pristanoyl CoA, it is a odd chain fatty acid. Right? Odd chain fatty acid. So what happens? Odd chain and the long chain. So what happens? It enters into the peroxisomes, carry out the oxidation, beta oxidation. Right? It will be converted into octanoyl CoA. Octanoyl is a 9-carbon compound. Right? And it enters after once the 9-carbon fatty acyl CoA is formed, it enters back into the mitochondrial matrix. Right? Again, the beta oxidation will be continued. Right? About a total of 6 cycles of beta oxidation happens with the liberation of acetyl CoA molecules. How many acetyl CoA molecules? We were having 19 carbon atoms, right? So 8 molecules, 12 into 8, 12 molecules of acetyl CoA, right? So 12 into, it's not 12. The 6 cycles of beta oxidation happens. So 6 cycles of beta oxidation results in 12 number of acetyl CoA and what? 1 molecule of propionyl CoA. Clear? So that is how the oxidation happens. Alpha oxidation happens. So a total of 6 cycles of beta oxidation. 6 cycles and the oro cycle, and then each cycle contains release of acetyl CoA molecules. So 12 molecules of 12 carbon atoms. Right? 6 cycles പറയുമ്പോൾ 2 carbon വെച്ചിട്ട് പോകും. Right? അപ്പോൾ അവിടെ തന്നെ 12 molecules of acetyl CoA form chedu. Then the remaining is released in the form of what? Propionyl CoA. Right? So that is how the fatty acids which are having side chains on the beta position is processed by the body. Right? So there are some the additional steps that I have included in my notes. But I think you follow this pathway only. You do not have to write the pathway which is written in my notes. It's something complicated. MSc level. You just write it down. The alpha oxidation of this particular slide. You write it now. So phytol is converted into phytanic acid. Phytanic acid is converted into 2-hydroxyphytanic acid. 2-hydroxyphytanic acid then undergoes decarboxylation and oxidation, give rise to pristanic acid. Pristanic acid is now converted into acyl CoA synthetase enzyme, so that pristanoyl CoA is formed, which is a C19 fatty acyl CoA. So C19, which enters into the beta oxidation and undergoes fatty acid oxidation, usual beta oxidation, and ends up in the formation of acetyl CoA molecules and propionyl CoA molecules. Clear? So that is how the complete oxidation is happening. Now, what happens if the alpha oxidative enzymes, the hydroxy fatty acids, are produced? What happens? What is the significance of alpha oxidation? These fatty acids, pristanoic acid, phytanic acid, etc., are very much important in the formation of certain specialized products like nervonic acid, cerbronic acids, etc. All of them, cerbosides, sulfatides, etc. All of them are important compounds which are present in the central nervous system. So all these substances are produced from the alpha oxidation, or odd chain fatty acid produced upon the decarboxylation pathway is used for the formation of sphingolipids, which is undergoing beta oxidation to form propionyl CoA and acetyl CoA. So the number of acetyl CoA molecules depends upon the chain length. Propionyl CoA is converted into succinyl CoA and then they enters into TCA cycle. So the one of most important significant things regarding the alpha oxidation is that the hydroxy fatty acids, that is, which are the hydroxy fatty acids? There's phytanic acid, 2-hydroxy phytanic acid. So these fatty acids are very much important for the formation of cerebronic acid, nervonic acid, etc. They are very much important for the proper functioning of the central nervous system. Apart from that, the odd chain fatty acids produced are utilized, and the propionyl CoA which is produced is converted into succinyl CoA, enters into the TCA cycle. Right. So these are the importance of alpha oxidation. Now, what are the clinical conditions? So the main importance is that alpha oxidation is running for converting the methylated fatty acids. The methylated fatty acids. The fatty acid is methylated at various positions. So methylated fatty acids they are subjected to digestion by means of or catabolism by means of alpha oxidation. Then they produce cerebronic acid. A cerebronic acid is responsible for the synthesis of cerebrosides and sulfatides. Clear? That is very important. Glycosaminoglycans formation brain development important materials molecules cerebrosides and sphingosides. Again sulfatides, etc., all of which are very much necessary for the proper development of the central nervous system. Then they also results in the production of odd chain fatty acids. Odd chain fatty acids the prostanic acid is an odd chain fatty acid. Odd chain fatty acids again, they can be utilized for producing acetyl CoA molecules and propionyl CoA molecules. Propionyl CoA is not wasted out. It is again entering back into the TCA cycle by getting converted into succinyl CoA. Now, what are the inborn errors related to this? The most important one is the infantile refsums, or it is the adult refsums disease. There are two types of refsum disease. It is the infantile refsum disease and adult refsums disease. So what is refsums disease? It's a neurocutaneous syndrome characterized by the accumulation of phytanic acid in plasma and tissues. Why is the accumulation happening? Because the alpha oxidative enzymes are defective. Clear? Phytanic acid ചെന്ന് കഴിഞ്ഞാൽ അതിനെ process ചെയ്യണമെങ്കിൽ എന്ത് വേണം? Which is the enzyme required? The phytanic acid, hydroxylase, oxidase, etc. So if they are deficient, what happens? Their processing will not be happening, so that they get deposited in the neurons. Right? So patients are unable to degrade the phytanic acid because of the oxidase enzyme, which are catalyzing the first step of alpha oxidation. As a result of which, what happens? Polyneuropathy, cerebral ataxia, mainly it affects the eyes in a main effect here, again central nervous system also, right? So there again, apart from that, cutaneous mode manifestations can also be seen. Cutaneous means skin manifestations, that is the skin becomes rough, dry and scaly. Then retina proper function because of the deficiency, or the phytanic acid is not getting oxidized. So symptoms are evolved slowly from the childhood throughout the adult stage, right? So patients, how can it be treated? Patients should avoid taking the green vegetables and milk. That's only treatment. So that's it. Then another the conditions, the infantile refsums disease. Infantile refsums disease is seen in Zellweger syndrome. Zellweger syndrome is associated with infantile refsums disease. So there, what happens? The phytanic acid is directly toxic. Hence, it gets accumulated and it gets deposited in the Purkinje fibers. And the accumulation of phytanic acid can result in the development of reactive oxygen species. And again, all those gets deposited in the retina, affecting the vision. Right? Hence, neuronal cells and retina are rich in why retina and neurons are getting affected, because they are rich in mitochondria, and they are the tissue which are affected in refsums disease, because the Purkinje cells Purkinje cells the alpha oxidation is primarily happening. So Purkinje cells are those cells which are present in the neurons. Cerebellar neurons specialized cells are Purkinje fibers. So Purkinje fibers these phytanic acids can get deposited and can cause neuronal problems and vision problems. Okay, so that is the conditions related to alpha oxidation, mainly refsums disease. Refsums disease are of two types: infantile and adult. Adult polyneurotic, the neurocutaneous syndromes. affecting the central nervous system and also the skin, and also affect the vision also. In case of infantile Refsum's disease, it's almost similar to that of Zellweger's syndrome. Okay, then peroxisomal oxidation also getting affected because the phytanic acid is not entering into